Hello and welcome to everyone. I am Luigi Crisci, PhD student at University of Salerno. In this presentation, I will be talking about our work called Toward the Portable Drug Discovery Pipeline with SYCL 2020, where we will share our experience on porting the ligand drug discovery pipeline to SYCL 2020. First, I would like to thank my colleagues from the University of Salerno, Macit Salimi Beni and Biagio Cosenza, then Niccolò Scipione, Davide Gadioli, Emanuele Vitali and Gianluca Palermo from Politecnico di Milano, and Andrea Rosario Beccari from Dompe Farmaceutici. In this talk, we will present an overview of ligand, an ex novo virtual screening framework owned by Dompe Farmaceutici. We will first introduce the concept of virtual screening applied to drug discovery, and we will review the ligand pipeline. Next, we will discuss the porting process of a ligand from CUDA to SYCL 2020, aligning the application structure and how the SYCL 2020 features allowed us to port the CUDA feature in a portable way. We will also present some experimental evaluation. We will show a preliminary comparison of two ligand versions, respectively based on accessor and unified shared memory. Next, we will show a performance evaluation study performed on NVIDIA V100, AMD MI100, and Xeon processor. Finally, we will conclude the presentation with remarks and consideration about the use of SYCL for industrial application. Virtual screening is the initial stage of the drug discovery process. The fundamental object that virtual screening manipulates is the ligand, a small molecule with usually less than 100 atoms. Here in the picture, you can see a simple ligand, in this case an arachidonic acid, where white sticks represents hydrogens, blue sticks represents carbons, and red sticks represents oxygens. The mesh structure instead represents the molecule surface. We can imagine a ligand as a graph, where a vertex represents an atom, while an edge represents a bond between them. It's also interesting that using chemical reaction, it is possible to generate a very large number of virtual ligands. In the context of drug discovery, a ligand corresponds to a drug candidate. The other fundamental object that virtual screening manipulates is the protein, a molecule composed of one or more chains of amino acid residues, which are the protein's basic blocks. Proteins have tens of thousands of atoms, therefore they are much bigger than a ligand. A protein structure example, in this case a 3D representation of the myoglobin protein, is shown on the right. Proteins are an essential part of the organism and participate in virtually every process within a cell. They perform a vast array of functions within the organism, like catalyzing metabolic reactions, DNA replication. They provide structure to cells in the organism and transporting molecules from one location to another. In the context of drug discovery, they represent the drug target. So, virtual screening aims at finding ligands that have strong interaction with the target protein. In fact, a ligand can be bound, under some constraint, to an empty space of a protein to change its composition, and hence its behavior. If the protein is associated with a disease, such event can significantly change the outcome of it. Ligands selected by this phase then are usually forwarded to later stage of the process. In a nutshell, we can see virtual screening as a preliminary filter in a drug discovery pipeline. Virtual screening comes with a few challenges. We want to evaluate as much candidate as possible. However, the bond between a target protein and a ligand highly depends on the ligand 3D atom displacement on the target protein. This means that a ligand must first be docked next to the target protein, and then their chemical bond can be measured. In the bottom right picture, an example of a ligand docking to a protein is shown, where you can see how the protein changes shape. Another challenge resides in the number of docking positions. In fact, a ligand can change its shape by moving its atoms. In the top right picture, you can see two conformation states of the arachidonic acid. The left picture is the classic shape, while the right picture it has been unfolded. Here the graph structure in the two ligands is the same, but shape drastically changed. This means that for a single ligand we have multiple ways to dock it to target protein. This procedure is called dock and score. The docking phase can be viewed as a relaxed lock-and key model. The protein is considered a rigid body, while the ligand is considered a flexible body. Here, the ligand is docked on a set of identified locations on the protein, called binding sites or pockets. Those pockets are chosen by domain knowledge to reduce the number of positions on which the ligand can interact with the protein, leaving only the most probable ones to be analyzed. Here, because a ligand is a physical object, different rotations called poses are tested in order to find the best docking position. In the bottom figure, you can see in details the COX2 protein and one of its binding sites, with the arachidonic acid docked inside. After the ligand pose has been docked to the protein docking site, in the scoring phase, the likeness of the bond between the ligand pose and the pocket is evaluated. To do that, scoring function takes into account atomic interactions like VDW, Lennard-Jones, metal bond, hydrogen bond, and solvation effects. The ligand drug discovery framework is a de novo structure-based virtual screening software owned by Dompe Farmaceutici and co-designed in collaboration with the Italian Supercomputer Center Sineca and the Politecnico di Milano. The distinguished feature of ligand is that it has been designed and developed to run on high-performance computing architecture. It has been already used to find drugs against the Zika viruses during the Anthrax project and against the SARS-CoV-2 in the SYCL project. The power of the ligand approach relies in the fact that dock and score is clearly an embarrassing parallel task. Each molecule in a pocket and each pocket in a protein can be processed concurrently, with no dependency between them. In this picture, you can see a summary of the ligand virtual screening pipeline. A protein with its binding sites and a set of ligand binaries to be tested are input to the application. Here, the ligand dock component performs the docking phase, in which each ligand and each ligand poses is tested against every protein binding sites. Here, the most promising poses are then selected and are input to the ligand score component, which applies multiple scoring function to each couple ligand binding site to evaluate the chemical bond. In the end, the scored ligands are forwarded to the later stage of the drug discovery pipeline or serialized to the storage. Ligand is part of the Ligand project, which goal is to create and validate an industrial application for drug discovery in high-performance computing systems. In this context, the evolution of HPC architecture is leading towards specialization and extreme heterogeneity, including future exascale architectures. Because of this, Ligand makes of code portability one of its primary objectives. In this field, SYCL matches this goal perfectly, able to write performance-dependent code while keeping native comparable performance. Ligand was originally implemented in C++, then ported to OpenSCC and then to CUDA, which is the version that we took as a reference for the porting. The CUDA version is composed of 17 kernels, with those on-device functions. The kernel flow is quite simple, where most kernels have a dependency from the previous one. For this reason, minimal kernel overlapping can happen. The principal features found in the CUDA code are warp-level primitives, substituted with SYCL 2020 subgroups and SYCL 2020 subgroup primitives, custom reductions, substituted with SYCL 2020 reduction, and custom group algorithms, mapped to the corresponding SYCL 2020 group algorithms. In addition, the ligand score component makes usage of CUDA dynamic parallelism, which was removed without performance differences. In our porting process, we wanted to take advantage of the numerous SYCL 2020 features. In particular, we wanted to support both unified shared memory and accessor-based memory management, in order to find the best fit for our application. In addition, subgroup shuffle and collectives allowed us to write more efficient and coincide code. Group algorithms, instead, helped us to raise ligand efficiency and to improve performance portability among architectures. Last, reduction native support allowed us to remove lots of the boilerplate code present in the original CUDA version and improve performance. At the end, by using SYCL 2020 reduction and group algorithms, we removed more than 430 lines of code. Performance tuning has been performed at a different level, application and kernel. Application tuning is performed by an auto-tuning framework combined with scheduler, which is not included in this presentation, which distributes and balances work among different nodes. Here, the block size and number of iterations on most kernel is chosen, depending on the workload type. In the kernel tuning part, we mostly worked on improving local memory usage with classic technique, like blocking, and adapting and optimizing manually tuned kernel sizes for a wider set of architecture. However, we found out that some kernel was suffering from a high register pressure that reduced the tuning space. For this reason, we were able to manually check the best tuning configuration. In the end, we developed two ligand versions, one based on accessor and the other one based on USM, to measure the difference in performance of these two approaches. Let's now examine our performance results. Here, our experimental setup is presented. We choose as backend SYCL, a modern SYCL implementation targeting both CPU and GPUs with a focus on leveraging existing toolchains, and DPC++, an LLVM-based compiler project that implements compiler and runtime support for the SYCL language developed by Intel. We configured NVIDIA setup with an IBM Power 9 AC922 CPU, NVIDIA Volta V100 GPU with CUDA 11, and an AMD setup with an Intel Xeon Gold 5218 and an AMD ME100 GPU. Here we can see a view of how the time is broken down in ligand. It is clear that most of the application time is dominated by a single kernel, the optimized fragment kernel, taking up to 95% of the execution time. It is a quite complex kernel of more than 420 lines of code, iteratively called multiple times. It includes a lot of different features like multi-level reduction, subgroup primitives, and blocking. Also, this kernel suffers of a very high register pressure that limits the kernel size. HBScore is another interesting kernel because it takes 2.2% of the overall execution time, but this is mostly due to group sorting. Because of the lack of sorting algorithms in the SYCL specification, we had to write different sorting algorithms in order to choose the one that performs the best in this specific use case. Other kernels include a kernel to align a ligand pose to the protein binding site, called the align kernel, which also suffers from high register pressure, two kernels for the best pose selection, one for the aligned section and another one for the optimized section, four kernels implementing different scoring functions, and a set of utility kernels that mostly performs matrix rotation. In our experiments, we benchmarked the accessor and the USM version of ligand 3.0. The main difference relies obviously in the memory management. The USM version is similar to the CUDA version in that each buffer is allocated and initialized at the start of the application and then deallocated at the end. On the other side, the accessor version declares each buffer at the start, and then the initialization is managed by the runtime. To keep the comparison as fair as possible, the kernel implementation shows very differences, beside the one related to the different buffer types in the two versions. In our findings, the accessor version and the USM version achieve very similar performance on different platforms and backends. To understand why this happens, we started profiling both the accessor and USM version of ligand. In this picture, you can see a view from NVIDIA Insight System, a performance analysis tool for CPU and NVIDIA GPU, analyzing a run of the ligand accessor version of a Tesla V100 using DPC++. On top, the light blue bar represents the GPU utilization, while the dark blue bar represents the single kernel execution. In red, instead, the memory allocation copies are drawn. On the accessor version, memory allocation, copies, and free are scheduled during the whole application lifetime, optimized by the SYCL DPC++ runtime. In the second picture, instead, you can see the same run in the USM version. Because memory allocation copies are made at the beginning of the application, the runtime cannot distribute and optimize memory operation across the whole application, thus reducing the overall performance. Furthermore, we noticed that memory operation seems to be much slower in the USM version compared to the one in the accessor version, but further investigation are needed to understand the nature of this behavior. However, such overhead is negligible, as memory allocation and data movement are usually performed only once and then reused during the application lifecycle. Also, we noted no significant differences in kernel execution time between the accessor and the USM version of ligand 3.0. Lastly, we present the result that we get on performance portability. We measure the application device time on multiple architectures and devices. Our analysis focused on all the 17 kernels, which were mapped to SYCL parallel 4 with the application. For 14 kernels, we have an excellent performance portability. In particular, our portable SYCL code was only 7% lower than a manually tuned CUDA version that can only target NVIDIA hardware. But the SYCL version can also target AMD ME100 and Xeon. On the other hand, for 3 kernels, we had lower portability. We have performed a preliminary analysis of this kernel, but such slowdowns are still under investigation. However, we found out that for the HB-score kernel, our Bitonic sorting kernel was almost 10 times lower than the CUDA one, implemented using NVIDIA CAB library. We are currently addressing this gap by replacing our sorting kernel with a customized and more performant version. For the optimized and the aligned kernel, they suffer of high register pressure, which seems to affect the quality of the generated code and to drastically slow down the kernel time with respect to the CUDA version. This problem is hard to tackle with current implementation of ligand. In fact, we are working on a new version of ligand that changes the parallelization domain of several kernels, including the optimized and aligned, and that will drastically reduce the register pressure. So, to conclude, ligand 3.0 relies on SYCL to provide performance portability. All CUDA features were ported to SYCL and now run successfully on a wide range of hardware. SYCL 2020 features also reduced ligand code complexity. Reduction in group algorithms allowed us to remove more than 430 lines of code. We also developed two versions of ligand, one based on accessor and the other one based on unified shared memory, with very similar performance. We also studied performance portability on multiple hardware. We got excellent performance portability on 14 out of 17 kernels, with manually tuned CUDA only 7% faster than portable SYCL on an NVIDIA V100. However, we got issue with 3 kernels because of high register pressure, and the new ligand 4.0 pipeline will attack this problem, and customized sorting algorithms. We would like to thank all partners from the ligand project. This project has received funding from the European High Performance Computing Joint Undertaking and the Italian Ministry of the Economic Development. With that, I concluded, and thanks for listening.